Hi, I'm going to talk about Germine mutation with predisposition genes in pediatric cancer. The definition. The frequency of germline mutation in cancer predisposition genes in children and adolescents with cancer and the implications of such mutation are largely unknown. Previous studies have relied mainly on candidate gene approaches, which are by design limited.
When a parent is first given that information that their child has cancer, one of the first questions that comes to their mind is, why my child?
For many years, the scientific community reasoned that the genetic changes that cause pediatric cancer were simply unpredictable. Results of a new study by St. Jude Children's Research Hospital and Washington University, published in the New England Journal of Medicine, are changing the way we think about this question.
We pursued a study where we analyzed the genome, the normal tissue, from 1,120 pediatric cancer patients and asked, are there mutations in true cancer predisposition genes? And our finding was really very surprising. It was that 8.5% of children with pediatric cancer have a hereditary cancer predisposition. This really is a foundational study that raises the awareness to the field that there are patients that have germline mutations in their normal tissue. They're at increased risk of cancer, and we do need to start establishing exactly how to follow these patients. And so at St. Jude, we have initiated a new study. It's called Genomes for Kids, and we are going to do an unparalleled level of sequencing on children with pediatric cancer.
By identifying which children have cancer predisposing conditions, we will be able to better treat these children, not only for the current cancer, but hopefully use the information to detect cancers earlier, later in their life.
With a commitment to rapidly apply these genetic research discoveries to patient care, St. Jude Children's Research Hospital has established a new hereditary cancer predisposition clinic.
We believe in working with the entire family because when you think about genetics and genetic conditions, they not only affect the child with cancer, but they have the potential to affect siblings and parents and other relatives.
The efforts made at St. Jude to not only understand the genetics of pediatric cancer, but to apply those findings directly to patient care, have the potential to fundamentally change the way children from around the globe receive treatment.
Applications in medicine. To better determine the contribution of germline predisposition mutation to childhood cancer, we use next-generation sequencing, including whole genome and whole exome sequencing, to analyze the genomes of 1,120 children and adolescents with cancer. We describe the prevalence and spectrum of germline variants among 565 cancer-associated genes, with an emphasis on the analysis of 60 genes that have been associated with autosomal dominant cancer predisposition syndromes. We also review records of patients with mutations and those without mutations in these 60 genes for information on family history of cancer.
For us, but first, there's some important new research on children and who are ill and their families. And this was fascinating when I was reading this morning. More than 8% of children with cancer have unsuspected genetic mutations. What does that mean? So basically what they did was they tested all these kids who have pediatric cancer, and they found that about 1 in 10 actually also have what are called germline mutations, which means that they were inherited, that could be a susceptibility to developing further cancer in the future. And not just for the children, but potentially for family members too. And what they then did was they can and they will offer the screening to siblings and family members of the children who have these particular mutations. It's quite fascinating because basically it changes their level of care. If you know you have a genetic mutation for a particular type of cancer, you can screen for it. And one of the examples that was given is that some of these kids, these small children, have the BRCA mutation, which is a risk factor for ovarian and breast cancer. It may not affect them, but it could certainly affect a parent, for example. And St. Jude said that they are going to offer free testing to the siblings and parents of the kids who have these mutations.
Fascinating.
Very,
Hopefully
very
we'll have a
fascinating.
breakthrough.
Yes.
Methods. The 1,120 patients included in the study represented the major subtypes of pediatric cancer. Whole genome, whole exome, or both types of sequencing data were generated with the use of germline DNA for 599 and 466 and 69 patients respectively, as part of the St. George Washington University Pediatric Cancer Genome Project. A total of 565 genes were chosen for analysis on the basis of review of the American College of Medical Genetics and Genomics. Gene lists and the medical literature and were divided into 5 non-overlapping categories. Data analysis. The sequencing data were analyzed for the presence of single nucleotide variants and small insertions and deletions and for evidence of germline mosaicism. Germline copy number variations and structural variations were identified with the use of the copy number segmentation by regression tree and next generation sequencing and clipping reveals structure algorithms.
Hi, I'm Rob Gray. Today I'll be discussing our study, Prevalence of Mutations in Cancer Predisposition Genes in Patients with Pancreatic Cancer. Our lab is particularly interested in pancreatic cancers that are caused by inherited mutations in the BRCA1 and BRCA2 genes. These genes are famous for causing breast and ovarian cancer, but also increase the risk of pancreatic cancer by up to 7 times that of the general population. Identifying patients with BRCA mutations can have important benefits. Relatives should undergo genetic screening, and carriers of mutations may benefit from interventions like bilateral mastectomies and salpingo-oophorectomies. Also, emerging evidence suggests that pancreatic cancer is caused by BRCA mutations of a distinct genomic landscape, prognosis, and response to therapy. Clinical trials are currently underway investigating agents that specifically exploit an effective DNA repair pathways in BRCA-deficient pancreatic cancers. We don't know how much pancreatic cancer is caused by mutations in the BRCA genes, because studies to date have focused on cohorts selected for family history or ethnicity rather than population-based cohorts. Other genetic syndromes significantly increase the risk of pancreatic cancer. These include Lynch syndrome, familial atypical multiple mole melanoma syndrome, familial adenomatous polyposis, hereditary pancreatitis, Peutz-Jeghers syndrome, and Lee-Fraumeni syndrome. The benefits of identifying carriers of mutations that cause these syndromes is similar to that of the BRCA genes. The goal of our study was to define how much pancreatic cancer is caused by mutations in known predisposition genes. We sequenced 13 genes that cause the syndromes I just described in a population-based cohort of pancreatic cancer patients. We wanted to detect at least one mutation in any gene mutated in over 1% of pancreatic cancer. To achieve this with adequate power, we selected 290 of 708 probands with pancreatic cancer from the Ontario Pancreas Cancer Study. The OPCS is a population-based cohort that recruits all consenting participants with a pathological diagnosis of pancreatic cancer in the province of Ontario, Canada. To maximize the precision of our estimate of the BRCA mutation rate, we randomly selected patients from 3 strata based on family history of BRCA-associated cancers. The site DNA from all 290 probands underwent targeted next-generation sequencing. Our bioinformatic pipeline detected genetic variants in the pancreatic cancer predisposition genes. Rare, non-synonymous variants classified as either pathogenic, of unknown significance, or benign based on their impact on the protein, publicly available databases. Each position in the targeted genes was covered by an average of 354 sequencing reads. Most rare non-synonymous variants were of unknown significance. On average, each proband carried 1.2 variants of unknown significance. Longer genes had more variants of unknown significance, supporting a more benign role for these variants. We detected 11 pathogenic variants that were then confirmed with Sanger sequencing. Although most have history of cancer, none met the Amsterdam 2 criteria for Lynch syndrome. Only one patient had a first-degree relative with pancreatic cancer. The two available tumors from carriers of mutations in the mismatch repair genes were immunodeficient for their respective protein. 3.8% of probands carried a pathogenic mutation after accounting for the stratified sampling design. This corresponds to about 1 in 26 pancreatic cancer patients. 2% of probands had mutations in genes associated with breast cancer, and 1.4% in genes associated with colon cancer. The only characteristics significantly associated with
The pros. The germline mutations identified in this study may provide insights into the causes of cancer. Knowledge of their presence may influence clinical management by directing cancer care, enabling pre-symptomatic genetic testing of relatives, guiding family planning measures, and facilitating the institution of potentially life-saving measures for cancer prevention and surveillance. On the other hand, the cons. This study has several limitations. First, several subtypes of pediatric cancers were not examined. Second, we did not study the parents or relatives of the patients in the cohort, and hence could not assess whether variants were neo-associated with the cancer phenotype among family members. Issue applied in Ecuador. In this study involving 1,120 children and adolescents with cancer, we found that 8.5% of the patients had predisposing gene mutations. Sequence coverage exceeded 10 times for more than 95 of the coding exons and 20 times for more than 85% of the coding exons in the genes of interest, which was sufficient for genetic accuracy. In conclusion, germline mutations in cancer predisposing genes were identified in 8.5% of the children and adolescents with cancer who participated in this study.